Professor Steven Gill, thanks very much for coming to Fearsome to tell us a little bit about drug delivery to the central nervous system. First of all, tell us a little bit about some of the main challenges with delivering gene therapies to the CNS.
Well, firstly, thank you for inviting me along to meet all the team. So the challenges that we face in delivering gene therapies to the brain is that you have to get the appropriate dose in the right location. Now, the appropriate dose we found is quite high. It's in terms of viral genomes per centimeter cubed of brain, it's about 1 × 10⁹ to 1 × 10⁻⁻⁻, in that kind of region. So these are high doses. So if you deliver that systemically, you have to fill your whole body with what is a massive dose, so a highly expensive therapy, and it fails to cross the blood-brain barrier efficiently. So even some of the viral vectors that people have developed, which can cross the blood-brain barrier, the proportion that gets across from systemic circulation is very, very low.
Yeah.
So it might be, say, 1/100th of the concentration in the plasma gets across into the brain. That's better than nothing, clearly, but it's never going to reach the appropriate concentrations in the right tissues for many neurological diseases. So that's our biggest challenge, really, is how do we get it there? And how do we avoid the systemic effects of gene therapy? So gene therapies delivered directly into the brain, for example, have never killed anybody. But when they're delivered into the systemic circulation, there have been several deaths associated with liver toxicity.
Right.
So you might think, oh, it's much safer to deliver things straight into the vein in somebody. Actually, it's proven to be the most dangerous way of delivering a therapy.
Right.
So if you want to fill, say, 1 centimeter cubed of tissue in the brain, you'd have to fill the body up with 70,000 times that dose just to get that, assuming it went across evenly, but it doesn't. So it's just not. I don't believe it's feasible now, and I think there are huge challenges in making it possible in the future. And for most neurological diseases, you've got to be within a very, very tight range, and I just can't see that happening unless you do a direct delivery.
So intravenous delivery, for the reasons you've already described, is basically out and is never realistically likely to be an option.
I think people are working towards trying to find a vector that will do this and be highly targeted. And yes, you can increase the degree of targeting to get across the blood-brain barrier, but I don't believe you can get the appropriate dosing without causing systemic effects. I think that's going to be really quite a challenge.
Yeah. So give us an idea then of some of the methods that currently exist for...
Okay, so it would seem that if you can't do it systemically, well, the next path that people are trying is going into the spinal fluid that surrounds the brain. But that has its own challenges. So the idea is you put it into the spinal fluid and that it can then penetrate by diffusion into the brain substance. But of course, a human brain's like this, it's quite a big structure. And you're putting the drug into a fast-flowing system. So you're making, you've got 150 mils of spinal fluid and you're turning it over 4 times a day. So it's moving rapidly. Not only is it going out through little granulations over the surface of the brain, but it's also going out down the nerve roots. It's exiting through multiple pathways. And the CSF doesn't flow. So if you put it anywhere in the spinal fluid, it's not going to homogeneously fill the whole of your spinal, what's called the subarachnoid space. It's going to be very patchy. So if you put it into the ventricle, it comes out of the ventricle and then flows over the surface of the brain and gets absorbed fairly quickly, but very little goes down the spinal cord. If you put it into the lumbar spine, basically you've got a column of pulsating fluid. It's not flowing up and out, it's just pulsating away. And a lot of it's being drained straight through along the nerve root canals, right? So it's being drained straight into the lymphatic system peripherally. So, and if you put it into the cisterna magna, which is just under the hole in the base of the skull, if you like, where there's a cistern or a collection of fluid. If you inject it there, then some of it goes down and some of it goes up. But again, it doesn't mix freely. So you're necessarily going to have a high concentration in local areas, and high concentrations you need to get diffusion. But equally, those high concentrations locally may be toxic. And we know that a lot of neurological diseases occur because you overproduce a protein. And what gene therapies are doing is creating a protein. So if you transduce a cell with many viral genomes, then you may be overproducing a protein which actually will kill the cell.
Right.
And a millimetre away from that is not getting enough dose. So, again, I cannot see how, getting a homogeneous dose, well, it simply isn't feasible to do it going into the spinal fluid that surrounds the brain. It's not, it's not that safe. And in fact, you get 1/10,000th of the dose that you put into the CSF gets into the brain substance itself, and it goes mainly straight out into the liver, and you get 100-fold greater transduction of liver cells than you do of brain cells by going into the CSF. So I don't believe that's a safe way forward. I mean, there are some indications for direct CSF delivery, and this is particularly for treating things like cancers, because 8% of people who have cancers develop spread into the spinal fluid around the outside of the spinal cord and over the brain surface. And that's sort of 20-odd% of say women with breast cancer will get this. It's a horrible way to die. And so really palliatively, you would deliver chemotherapy into the spinal fluid, but again you face the same issues. It's not circulating. So how do you get an even concentration of your chemotherapy into the right place? That remains quite challenging. That's something we're working on right now, because if you put it in the lumbar spine, again, it'll concentrate around the nerve roots. It can be toxic to your nerve roots, and you end up with a secondary problem. So that's one thing. People are also trying to deliver antisense oligonucleotides for certain diseases, particularly spinal muscular atrophy. Now, this is a disease that affects children, where the permeability may be different to adults in terms of going from CSF into the spinal cord. Of course, a spinal cord in a child is very tiny, and you can get some penetration into the peripheral nerve roots and through the outer layers of the spinal cord, and that has shown some efficacy in that condition. But for other and the major diseases we're interested in, major neurodegenerative diseases, the Parkinson's, the Huntington's, the motor neuron disease, this is going to be a real challenge to do it via the CSF, and I just don't think it's feasible.
So for some of these diseases that you've just mentioned, what are some of the deep brain structures that you may need to try and access?
Right, so in many of the diseases you can actually pick out individual structures that are mainly affected by the pathology. So it's in Huntington's disease, the key structures are the putamen and the caudate nuclei, the part of the basal ganglia, but you also get general cortical changes as well. Now, it's possible by selecting the right type of virus, which is carried in particular directions along the axons, to target one structure, and it then feeds out, it's then carried to other structures. So you might deliver into, say, the thalamus, which is connected throughout the whole of the cortex, and deliver a drug or a therapy to large areas of the cortex. In Huntington's disease, you mainly target the putamen and the caudate, but those have connections with the cortex as well. So if you have a virus which will retrogradely go back up the axons, you can get to the cortex as well. So although by direct delivery, you may deliver to quite a small volume, you can actually have profound effects over wide parts of the CNS.
Can you tell me what the structure of the pons looks like?
It's really a little bulge on. So the brainstem, so you've got the brain on top here, and the brainstem comes down. This little bulge down the brainstem, so halfway down the brainstem, and it's where you get a conglomeration of fibers coming from the whole of the brain, many of them from the critical layer 5 of the cortex. They're going into this structure and then feeding back to the cerebellum, which is processing information to improve your accuracy of movement, your precision movements in particular. It's got a lot of cognitive effects as well. So, and through that area, you've got all the sensory fibers coming up and all the motor fibers going down. And you've also got brainstem nuclei. So in something like motor neuron disease, where you lose your bulbar function, your ability to swallow and move your face and do all those things, you could potentially put your gene therapy right into that location, treat all those particular nuclei directly, but also pick up the motor tracts going right the way down the spinal cord and up to the motor cortex as well.
So what would you see as the solution then, Steve, for targeting these sometimes interesting structures deep in the brain.
So what we're trying to achieve is a homogeneous dose of the virus carrying the transgene throughout particular structures in the brain. And it might be, say, for Parkinson's disease, the putamen, or for Huntington's disease, the putamen and the chordates, and so on. So this can be done by putting a very tiny cannula or tube into the brain and infusing your drug down that tube at a very slow flow rate to enable you to basically create a pressure gradient at the tip of your cannula. Then you can then drive that infusate through the tissue, through between the cells in the tissue, through the interstitial space, and you can carry by bulk flow these quite large molecules over the relevant distances. And that process or that method is convection enhanced delivery. Convection enhanced delivery has been used in the lab for many years, but translating that to deliver therapies to humans is a bit of a challenge, because you're going from, say, a mouse which has got a brain 3,000 times smaller than a human to a non-human primate which is 15 times smaller than a human. So you're delivering over these relatively small volumes. So i.e., if you deliver by a millimeter in a non-human primate, you've got to go 1.5 centimeters in a human, right? So these are big changes from the lab to the clinic. So the way this is done right now, with the only approved therapy for delivery by convection enhanced delivery in Europe, is being delivered by a system called the ClearPoint system. And this is a system where the patient is anaesthetized, scanned with an MRI, the target is defined, and the entry point is defined for each cannula. And the first thing you do is you mount a small stereotactic frame over the entry point, and then you have to move, if you like, a cylinder of fluid and orientate it so it's aligned with your planned trajectory. Take the cylinder of fluid out, make a hole, and then deliver your cannula down to the target. And one of the issues here is that the infusion has then got to be done kind of real time by infusing contrast agent with that, and then advancing the cannula down through the target to try and push the drug out over the volumes. So this is a very interactive process. The surgeon's constantly having to manipulate that cannula to fill relevant structures. And you have to do the surgery sequentially. So when you've delivered your therapy to one target, you're then having to put on another frame and then deliver one to another. So the whole process is very prolonged. And so for some conditions such as Huntington's disease, where you may have, in their case, 4 targets, both putamens and both chordate nuclei, you've got to do 4 individual procedures, if you like, and do them kind of sequentially to then fill these structures. And that means it's taking beyond 12 hours, often up to 18 in some cases, to deliver the therapy. And there's a tendency to really drive the drug very fast through the tissue to try and fill it.
So in contrast to that then, how long would a patient be in surgery with the NeuroChase system?
Okay, so to solve a lot of the issues that we see with current technology, the ClearPoint system, NeuroChase is very much focused on putting multiple catheters in very quickly and operate in a clean environment operating theatre, making the delivery very, very simple. So it's not dependent on highly skilled surgeons spending hours individually doing things. We want to make the delivery semi-automated so that there's assisted trajectory planning. The surgeon's just following a process. We're trying to take numbers out. So if you're communicating in an operating theatre and you've got a set for a standard stereotactic frame, 9 individual trajectories, there's lots of numbers you're juggling around with, and then each tool, if you're fixing things to the skull, then the distance from the skull fixation to the target has to be known for, how you're drilling the skull or creating shapes in the skull to fix your device to the skull. So there are multiple measurements that need to be made, and again, we developed a system where you take all those out, all those measurements and the handling of numbers in theatre out by simplifying the whole process. So that comes with the tooling. And it's now possible for us to put in devices in the skull within 10 minutes each. And so within an hour, we can get our 6 cannulas in for a disease such as Huntington's disease. Then we get the patient out of theatre. So the next challenge that we're trying to solve at NeuroChase is, okay, so you've got a patient with 6 catheters in the head, and each is going to receive a different volume of drug. And so am I going to now connect all these tubes up to multiple infusion pumps, 6 infusion pumps, and program each individual pump to deliver just that volume at certain flow rates? And how do I, am I ramping up the infusion? When's it going to turn off? It gets very complicated. And within a very short time, you're going to be in difficulty because somebody's made an error. So we have to make that whole process simple. So we've solved that problem, we believe, by pre-filling a dispenser in a clean environment, done by a pharmacist, checked by others before it's dispensed, gets onto the ward, and this vial containing this volume is for cannula number 1 and 2 and so on. And so somebody who might be a specialist infusion nurse would then connect them all up, 1 to 1, 2 to 2 and so on, and then those are connected. Each vial is then connected to an infusion line, which is a syringe pump just delivering saline. And you can turn all the pumps on at once, and it just drives the infusate down all of the individual lines to the individual dispensers. And in the proximal part of the dispenser is basically the fluid, the infusate that's coming, the saline. And that's in what we call a hydraulic chamber. That drives a piston to empty the distal chamber, the dispensing chamber. So when you turn those pumps on, it's going to drive that volume until it's fully delivered, and then the piston will hit the distal end of the dispenser. The pressure rises in the hydraulic chamber and then feeds back to the pump and turns it off. So you can turn 6 pumps on, all with different volumes, and they all deliver simultaneously, and each will turn off when it's delivered its volume. So that makes it very, very safe. There's nobody having to program anything. It's all done in a very standardized way. And again, you're not exposing people in the operating theatre to potentially biohazardous therapies. It's all being done on a ward by specialist people. And the theatre, you can be moving through the theatre very quickly. You can deliver your kit on this patient, they're out, and in comes the next one. And therefore, if you're going to treat the hundreds of thousands of people that need treatment for some of these diseases, you have to have that type of system which is very safe, and you've really thought about how you can avoid human error. And surgeons are surgeons, and they just want to put the kit in, and people who can monitor patients, maybe neurologists or neurological registrars, are overseeing the infusions, or specialist nurses, and they're doing their own thing which they have particular expertise in.
So this is obviously brain surgery, Steve, which is your trade, but to me that sounds kind of daunting.
Yes.
How has NeuroChase solved some of the safety concerns that someone might have around that type of surgery?
I think, almost more than any other type of surgery, we have an unprecedented level of precision in what we're doing. We have images of the brain which are incredibly high resolution, so we can see tiny, tiny vessels. So if you're doing bowel surgery or something, everything's on the move, and you can't see any of the detail that we would see in the brain. And the brain is fixed in a box. And the box, the skull, can be fixed relative to your delivery system. And that can be a robot with sub-millimeter precision. And we can now have software that avoids blood vessels and so on. And what we put into the brain is absolutely minuscule. We don't even remove the hair. We just make a tiny incision. We put in, the cannula itself is 0.5 millimeter in diameter, right? And then there is a tiny guide tube that's just over 1 millimeter in size. And these are tiny things. And so we can make sure we can deliver these things very efficiently in a short time, and that's also important. So we use the technology. It's not down to free-handing. The surgeon never does anything freehand. It's always done in a highly precise way. And even when the patient gets on the operating table, if the brain was to shift inside the skull, we can determine that to sub-millimeter levels of precision, because we take an image after injecting some contrast into the bloodstream, and we can see where every blood vessel is next to your cannula. So everything's adjusted around where those blood vessels are moved to. So this is really unprecedented in surgery, our accuracy. And when you know what you're doing, and you're just being guided by, say, a robot, which can set that trajectory absolutely precisely, then this is incredibly safe in terms of the particular risk to the patient. Of course, nobody wants brain surgery, but things have changed, really driven by technology now. So this can be kind of day case surgery in a way. It's the patient goes to sleep, they have tiny incisions and very, very tiny devices very precisely placed in the brain. So for some time people have been putting things like recording electrodes if you have epilepsy to find out where the epileptic focus is. People put up to 12 electrodes into the area where that might be. And the risk of 12 cannulas going in, which are much bigger than our device, is overall low. So there's something like a 1% risk of those 12 cannulas all going into one area without the level of precision of guidance that we can achieve now. There's a 1% risk of bleeding. And the chance of that actually causing a neurological problem is very low, under 10%. So 1 in 1,000 people are having a problem from big things going in the right place. We're in a totally different world now in terms of our precision and our ability to put tiny things in.
So the NeuroChase catheters are actually much smaller than a normal deep brain stimulation electrode, for example.
They're 0.5 millimeter in diameter. And so, whereas deep brain stimulating electrode is something 1.3 millimeters in diameter. So that device going into the critical structure, it's many-fold smaller.
Wow.
And so that's what's taken these years for NeuroChase to develop. We know we can deliver those with unprecedented accuracy.
Yeah. Now, some people are using ultrasound, I think, to try and bypass the blood-brain barrier. That, to me, kind of sounds nice, sounds non-invasive. Tell me a bit about that.
So the idea here is you inject micro bubbles into the bloodstream, and you focus your ultrasound around the volume of your brain tissue you're interested in. And these are normally fairly small volumes that you can focus on. Maybe, say, a centimetre cubed is about the size you get. And by sending pulses into that area, you can burst the bubbles, these micro bubbles, as they're flowing through. And that can fracture, make little holes, if you like, through the blood vessel membrane, the basement membrane. And that allows drugs to permeate into your brain tissue through those tiny little holes. Now that sounds pretty good, but it's not just your drug going through, it's also your immune system going through into the tissue. And what goes in also comes out, right? And you have to fill your whole body with, you're treating 1 centimetre cube, you filled 70,000 centimetre cubes of your body with your viral vector. So your doses are still huge. You're still filling your liver up with your gene therapy.
Yeah.
And that's where the toxicity occurs. So, and also now you've got the problem is, how do I treat these critical volumes? So you've got to hit multiple targets with a very complex system, which is very expensive. So so the patient has to go in, have their head shaved. They put pins, hold the head in the right location, and put a great big thing over their head with fluid and things. And I wouldn't say it's non-invasive. I mean, you're invading with enormous amounts of energy, highly focused. So there are risks around that. Yeah. You can get inflammation occurring and so on. So the technology improving, but again, this is not something that happens quickly. It's something it's a lot of planning. There's a lot of complexity around it. Mm-hmm. And are you still going to get your 1 times 10 to the 10 viral genomes per centimetre cubed homogeneously in that volume? No, you're not. Mm-hmm. It's going to be, the dosing is huge. Now, the thing about delivering directly into the brain tissue is we can control exactly where that's going to go. Mm-hmm. And once it's in, it can't get out. So, even if you put chemotherapy into the brain directly by convection, it doesn't get out very easily because the blood-brain barrier is working in your favour to keep the drug there. So you have very prolonged exposure of a drug once it's in there. And, it's a quick procedure and you can monitor exactly what you're doing as you're doing it. And you can also get our cannulas out under local anaesthetic, so you don't have another anaesthetic to take them out. They come out with tiny little screwdriver things, you can put them out. So that's done in a sort of a clean room on a ward, for example. So I don't think filling your body up with a drug and then using ultrasound, these very, very expensive machines, is going to get you to treat the tens of thousands of people we need to.
Yeah. So thanks, Steve, for coming to see us today. It's been absolutely fascinating finding out all about the challenges that exist with getting gene therapies into the central nervous system and the incredible ways that NeuroChase is solving those problems.
Well, thank you, Alan. It's been a pleasure to come and see all your lovely technology and things. It's been great to meet everybody. Thank you.